Tetralogy of Fallot, a form of congenital heart disease defined by an anteriorly malaligned ventricular septal defect which leads to a subpulmonary valve obstruction, affects 1 in 3,000 live births. While there is a well-known heritability risk in Tetralogy of Fallot patients, the genetic underpinnings of the disease remain unknown. In this manuscript by Merriam-Reuter et al., the authors re-evaluate data from the largest genome-wide sequencing dataset of 811 probands with Tetralogy of Fallot in order to identify rare, likely pathogenic variations in genes associated with congenital heart disease outside of ones found in previous studies. This reanalysis showed 5.9% of the probands had at least one likely pathogenic variant in a congenital heart disease associated gene. Two genes, FLT4 and NOTCH1, comprised of half of these variants. In 16 individuals, the authors identified loss of function variants in 3 emerging congenital heart disease candidate genes, KDR, IQ-GAP1, and GDF1, all with a variant prevalence significantly enriched in the Tetralogy of Fallot cohort study when compared to the general population. The findings in KDR and IQ-GAP1 corroborate previous studies, suggesting them as a Tetralogy of Fallot and congenital heart disease gene. The authors also found evidence of a highly interactive network of encoded proteins, with 88.5% of the newly discussed genes connected in this network closely tied to the central nodes of KDR and NOTCH1. Novel results from the study underscore that the heterogeneity of Tetralogy of Fallot may have some overlapping molecular pathogenesis, identifiable through analysis of the exome landscape of the disease. This may ultimately inform potential therapeutic targets.